Swayam Prabha. Digital India. Educated India.
Welcome. So we have been discussing neuronal encoding and decoding, and we ended the topics on neuronal encoding and decoding, that is the model based understanding of the forward problem, that is going from the stimulus to the neuronal responses, or from the neuronal responses to the stimuli, and that sort of ended with the discussion on maximally informative dimensions, which sort of is the most sort of advanced way of looking at systems approaches in understanding neuronal encoding and also decoding. So what often happens is that we won't be able to use all kinds of stimuli, and we are limited experimentally to span the entire stimulus space in terms of presenting stimuli and getting responses for all kinds of stimuli, which is physically not possible. And so we are sort of limited in that sense using the model based approaches. Further, there may be cases where the stimuli are not parameterizable at all. For example, as you already know, in the case of olfactory and gustatory stimuli, the stimuli are discrete sort of elements in discrete space, and so it is not possible to parameterize the stimuli and hence model based on the stimulus parameters and obtain the responses from there. So even if in the case of auditory, somatosensory and visual stimuli, while at the basic level the stimuli are parameterizable, when we go up to the level of objects, let's say a visual object, it is not that easy to again parameterize those objects. Even if we talk of speech, or let's say if we are studying species specific vocalizations, it is not that easy to even define an object in the auditory world and also to parameterize the whole structure as a parameterizable element. Because there may be many types of sequences, let us say, for example, as in speech or in the visual system, there can be many different kinds of objects that are surely at one level they are parameterizable, but maybe at where we are looking at the encoding of those objects, it is not possible to actually think of responses based on those pixel values, if you will, in the case of an object. So it is not like the sum of parts give a whole, or some function of the parts give a whole, some simple polynomial function of parts give a whole. So in such cases we are at a loss if we try to go with the systems approach. So in those cases where, if depending on the question that we want to answer, if we have a certain set of stimuli to work with, which are distinct objects, let us say A, B, C, D, E, or what have you, then we can think of neuronal encoding or even decoding in terms of discriminability. So we will now start our discussions on neural response based discrimination of stimuli. So in other words, what we mean is that let us say we have a neural system and we are recording the spike trains that is, let us say response 1 is a certain spike train in response to stimulus 1, and if we give the stimulus 2, we get the response 2, some other spike train. Now the question is that can we say that the neuron, based on these two responses, or many repetitions of R1 and R2 or stimuli, and hence many replicates of R1 and R2, based on that, can we say that the neuron encodes these stimuli? To answer that question, we ask the question that whether based on the responses we can separate out the stimuli or not. Well, we are giving a very simple example where we have only two stimuli. This can easily be, these ideas can easily be extended to many number of stimuli, and depending on how well we can discriminate the different stimuli based on the responses, we can say that the neuron has a high fidelity of encoding these stimuli. If the responses are very different for all the different stimuli, we can obviously then say that okay, the neuron, based on these responses, since we can tell the stimuli apart very well, that means the neuron provides information about the stimuli. We are not providing a model in this case where we are transforming from the stimulus space to the response space, but indirectly saying that there must be some model there which allows the formation of very different kind of responses in terms of spike trains, and hence the neuron is actually encoding these stimuli. So if as you may well understand here, that if for two stimuli or three stimuli the responses are statistically identical, that barring the stochasticity in repetitions of stimuli, the responses are basically identical, then that means that based on those responses we cannot tell the stimuli apart, and hence the neuron is not encoding those stimuli. So this is sort of the basic understanding of this approach of understanding encoding and decoding based on discrimination. Now a very important point here is that we will allude to here is that okay, let us say for stimulus 1 we have a response that is a spike train. Let us say in trial number 1 it produces a single spike here, in trial number 2 it produces a single spike here, in trial number 3 it produces a single spike here, exactly at the same time post the stimulus onset. Every trial it produces a spike at the same particular time point after the stimulus on. Let us say for the case of stimulus 2, now we have again, let us say in trial 1 in response to the stimulus it still produces one spike, but at a different time now. Let us say this is the duration of the stimulus in time, in trial 2 again at the same time, again in trial 3 again at the same time. So essentially what we are saying is, if white is for stimulus 1, then cyan is for stimulus 2, here the two different cases. So this is the spike due to stimulus 2. So if you look at this simple example with the 3 repetitions and 2 stimuli, we based on what we are seeing, we can obviously say that yes, the neuron actually discriminates the 2 stimuli because the spikes are occurring repetitively at the same time, and in the other case it is repetitively occurring exactly 100 milliseconds or 200 milliseconds later. So the actual answer to this question is that so if we now ask that okay, so what makes the neuron, what makes the neuron, what actually decides whether the neuron discriminates the 2 stimuli or not? we, by looking at those spikes at a particular time for stimulus 1 and at a delayed time for stimulus 2, we can easily see that they are different. But different to our eyes is not necessarily different for the element to which this actually matters. And that means actually the neuron that we are recording from is actually projecting this information based on its axonal projections to some other neuron. And now that some other neuron needs to be able to respond in different manners to the two stimuli. So, in this particular example, we will see that the next neuron may or may not have a different response in the two cases, which we will see in a little time. So, it all depends on how the next neuron treats these spike trains. So, whenever we are talking of discrimination, there are approaches where we will talk about an ideal observer kind of scenario, where with no assumptions, if an ideal observer which can encode or rather decode all the information present in the spike trains coming in, whether that can differentiate stimuli or not. So, this is based on the ideal observer analysis. There may be assumptions in those analyses about the nature of the spike trains, the statistics of the spike trains, and so on, or there may not be assumptions. There are approaches where there aren't any assumptions also, and we say, okay, that based on that so much discrimination would be possible. So, it is not necessary that a postsynaptic neuron that is being provided this information by the neuron that we are recording from, that may or may not be able to discriminate it. That may or may not be able to reach that level of discriminability that an ideal observer would. So, other than the ideal observer, so the ideal observer approach based on the scenario can sort of be an upper bound as to how much discrimination is feasible. But in reality, we are left with biologically plausible discrimination. That is, we say that biologically plausible methods of decoding, basically. So, if we can think of neurons that can actually respond in two different ways for the two different spikes, the white and cyan, then we can say that, okay, if the next neuron behaves in that manner, then we can differentiate them. But if the postsynaptic neuron is such that it is unable to respond in a different way to the two spikes, that is the white spike and the cyan spike, then we cannot discriminate them. Then the stimuli cannot be discriminated. So, even though the neuron is producing different responses to the two stimuli, and possibly essentially in that sense encoding the stimuli, but the recipient neuron, the receiver of this information, is unable to utilize it. So, we often, we will go into this discussion later on also in this part of the course, where we will be using this biologically plausible postsynaptic neuronal decoding, based on which we will say whether the neurons responses can be used to discriminate the stimuli or not. So, that's the second important thing. And finally, an even more possibly important thing is the causality aspects of things. So, even if, let us say, this is S1 and this is S2, and this is producing response 1 and this is producing response 2, and let us say with a biologically plausible methodology, we are able to say that if the postsynaptic neuron is has these certain properties, then it would be able to discriminate between R1 and R2. And let us say in reality also this neuron projects to a neuron which has exactly those properties that we assumed that the postsynaptic neuron needs to have in order to discriminate R1 and R2 or differentiate based on R1 and R2. It is still not necessary that this is actually being used by the organism to differentiate the stimuli. So, if, up to this particular point, even if it is the decoding is biologically plausible, even then this whole thing simply may be correlational. That is, the responses are different because the stimuli are different, but it is simply just a correlation based observation, and the organism doesn't use this information at all in order to discriminate the two stimuli. Maybe the organism or the animal doesn't even discriminate those stimuli. So, in order to establish that aspect of causality, it is not just this idea is not just applicable to discrimination studies, but can go on to and can be used for the rest of the things that we have been talking about, but it is appropriate to discuss this here. So, we have to, along with these analysis, we have to take help of empirical methods, again experimental methods, to establish causality, in the sense that can we behaviorally alter or can we have an animal or I mean an organism, whichever we are studying, can we read out the discrimination behaviorally, and let us say that the animal or organism is able to discriminate between S1 and S2. So, it can tell apart S1 and S2 with some behavioral readout. Now, from the neuron that we are recording, we have responses R1 and R2, and our hypothesis is that it is because of this R1 and R2 that the stimuli are being discriminated. So, what essentially we need to do is now manipulate the response to the stimulus S2 in some way, and maybe make it like R1, and see if the animal cannot discriminate S1 and S2 anymore. This is in an ideal world I am talking. Similar experiments are done, but not at the not the way that I am currently talking about, not so precisely at the single neuron level yet, but we are getting there. And so now again, when we play the stimulus S2, we change the response or manipulate the neuron's activity to make it like R1, and see that the animal sees this as the stimulus S1 and is unable to discriminate between the two stimuli. Again, when we play, and reversibly, again when we place a present stimulus S2 and let the response be itself, be R2 itself, the animal is again able to discriminate S1 and S2. So, this way we can actually establish the causality of this whole encoding decoding process. That is, reversibly change and manipulate the responses in order to test the hypothesis about whether the responses are being used by the organism or animal to interpret these stimuli or discriminate these stimuli. So, while of course this is not really a part of the computational neuroscience domain, but since you are learning to be um, to to be neuroscientist per se, I mean, definitely if you do computational neuroscience, you need to also know the basic neurosciences as well, and this aspect of causality is extremely important, because based on correlational observations we can conclude many things which need not necessarily mean much, and so it's important for you to also realize those things. While computationally you approach a problem you based on correlational observations and you make a model, but it may turn out that it is of no use actually. I mean, I wouldn't say no use, but it is not what the actual animal or what is actually happening in the brain. So that is a word of caution that you must always keep in mind, that not necessarily that correlational observations are the end of it. You also have to look for causality, that is the observations you are basing your results on need to be more established than just correlational observations. Okay, so in order to approach now quantitatively the discrimination of stimuli, so we first start with the idea of the response measure. So as we have spoken earlier, we will always need to define our response measure, that is based on which we will be talking about discrimination being done by the neuron or the postsynaptic neuron. So usually the most used and is actually and also observed that neuronal rate responses often corroborate our behavioral observations. It's, I mean, not all the time, but a lot of examples are there where cortical neurons, at least, their rate responses correlate well with behavioral measures, and so most often scientists have used response rates over a period of the stimulus as the response measure. And so once we have response rates, given our previous discussions on statistics of responses, if the rates are high enough rates, high enough rates implies that they are distributed Gaussian. That is, given that originally they are Poisson in nature, Poisson distributed. Now if we take those leaps, that is, we have random time windows based on which we are computing rates from spike counts. The spike counts are Poisson distributed. So let us say spike counts are Poisson distributed, and now with random window sizes, if we convert the spike counts into rates, we get a continuous random variable now, not discrete like the spike count with a constant window size. And if the rates are high enough, these two together, then we know that it approaches a Gaussian distribution. And so based on this Gaussian distribution, the measure that often comes about to quantify the discrimination is based on, let us say, this is the distribution for one particular stimulus S1. So this is Gaussian in nature. Let us say this mean rate is mu1 and this variance is sigma. And similarly for stimulus S2, the mean rate turns out to be mu2 with equal variance like this, and the standard deviation is sigma. So both have equal standard deviations or variance sigma square, and the distributions are Gaussian in nature. So we often quantify the ability to discriminate the two stimuli S1 and S2 based on these rates using the measure called D prime, which is simply mu1 minus mu2 divided by sigma. That is the difference in the mean and normalized by the standard deviation. And alternatively, if the variances are different, then it is usually corrected to be mu1 minus mu2 by square root of sigma 1 square plus sigma 2 square, and depending on the situation, a multiplication by square root 2 is also done. So using these methods now, in very early studies of discrimination, where these kind of analysis were done, this D prime measure was used, and it was generally considered that a D prime of 1, D prime equal to 1 or more, absolute D prime of 1 or more, is where the discriminability of the two stimuli starts. That is, if the rates are distributed more than if the rates are distributed such that the means are separated by more than standard deviation, then based on those rates, an ideal observer analysis, we just start to differentiate the two stimuli. With D prime less than 1, we generally consider that we are unable to discriminate the stimuli. So that is I mean that number D prime equal to 1 is sort of a criterion. You can always have D prime to be 2 to be your criterion, which would mean that you are more accurate in terms of discriminating the stimuli. So that criterion can be in your that criterion is actually not the critical point. The point is that when we are comparing discrimination between two stimuli and another two stimuli, we can always say if the D prime is greater than the other one, then we are better discriminating the one with the greater D prime, the pair with the greater D prime more than the other pair. So and whether they are being discriminated or not, that will again depend on the criterion. So this one measure D prime, it requires quite a few assumptions here, which is that we use the idea that okay, the rates are distributed Gaussian, which need not be true. It also may be that the variances, even though they have variances, the estimates are improper, and most importantly that the distribution is not necessarily Gaussian. And so we need such measures of discrimination where we do not need to make those assumptions about the model, about the statistics. While this is okay, the Gaussian for high enough rates is quite okay, but there will be many situations where that is absolutely not true, and so our D prime measure is going to fail, and so we require measures that are independent of such assumptions. And as we saw, to go to measures independent of assumptions, we again take help of information theory, and the way to use information theory for discrimination is a question for you, if you think about it for a minute, if you recall what we discussed in information theory, what sort of measure can be used for discrimination, you may recall that we had a measure between two distributions statistically, without making assumptions about the underlying distribution, and that is the Kullback-Leibler distance, or KL divergence, or relative entropy. So KL distance can be used as a measure of discrimination without any application of assumptions of the statistics or nature of the distribution of the responses. So these kind of measures, while very useful, again has the associated problems of estimating them correctly. That is, we require large amounts of data, and we will also require that the correct estimation, that is removal of bias, which is a problem with such information theoretic measures. So in the next lecture, we will be starting off our discussion on discrimination based on KL distance, and from then we will go further, as we have discussed, with biologically plausible decoding methods and look at discrimination of stimuli. Thank you.